Yeah, if you can get closer to the microphone, thank you.
Morning, everyone. Thanks so much for coming, and welcome to our seminar on this beautiful, nice, rainy and wet morning. I'm just going to introduce our wonderful Jody Oliver, who is going to be presenting today. A bit of background: Jody did her MSc at University of Johannesburg, and she completed that in 2018. And she's since worked for a short time at the Marine Invertebrate Collections at the Eco Museum. She's worked as a research assistant at the University of Johannesburg and at SAIAB as well with Dr. Kerry Singh. And I first met Jody in 2020 when Kerry and Jody came up to SAIAB to discuss a project proposal to do with coelacanths and fish and eDNA, really cool stuff. And they were wanting to know if I wanted to collaborate. Of course, no brainer. I was in. And that's where it all started. So in 2021, that project got funded. And Jody signed up to do her PhD. For better or for worse, I'm one of her supervisors for Jody. And it's been an absolute pleasure, the whole project, and in particular Jody. Over the course of her MSc and her PhD, she's had 6 publications already out. The latest one just last week, and we're going to be talking about that. Another one is already submitted. We're just waiting for reviewers back, and a couple more in the pipeline. So if anyone wants to know what a hardworking, excellent work ethic is, look no further than Jody, workhorse of note. In 2024, as all of you here at SAIAB know, she joined the Aquatic Genomics Research Platform team and has been invaluable in helping me run the platform upstairs, and is doing her PhD part-time. Again, excellent work ethic. It can be done. And what she's going to do today is just going to talk about a little bit about her PhD, not a whole PhD, just certain aspects of it, and the exciting journey she's been on. So without any further ado or fanfare or anything, I introduce Jody Oliver.
Thanks, Gwyneth. Good morning, everybody. Good morning, everyone online. So yeah, like Gwyneth said, I'm basically going to tell you guys a story over 4 chapters about the rich life beyond the surface. So all stories has a beginning, right? And ours started with a dream. So this dream, of course, was fueled by the ASAP open call, and this was back in 2020, in the thick of COVID. And basically, at the time, for those of you that don't know what is ASAP, ASAP is the African Coelacanth Ecosystem Program that was initiated back in 2000 when the coelacanths were found in northern KwaZulu-Natal. And basically, this program was one of the first multi-disciplinary and multinational research program that basically facilitates or supports ecosystem research in the Southwest Indian Ocean. And basically how they do this, today there is an array of research vessels with skilled personnel, like skilled and amazing personnel, by the way. We have the remotely operated vehicle over here that is flown, I guess you fly RV, flown by Ryan Palmer. And then we, of course, have lots of other equipment, cutting-edge equipment, such as your CTD with Rosette, that has, you can do oceanography data, you can do water collections, and this is basically just the tip of the iceberg. There's so many other cutting-edge equipment that you can use. And of course, we also have in this very institution, we have the Aquatic Genomics Research Platform, like Gwyneth mentioned. She is the platform manager, and basically we house next generation sequencing technologies, such as your Illumina MySeq over there, and then we have the two Oxford Nanopore technologies, which is your Mark 1C, and then we also have a P2 Solo. So we have all these cool and amazing toys to play with, and to really cutting edge research. So of course, with all that said, at the time I was a research assistant at SAIAB in the marine program, and Professor Kerosene came into my office and she said, Jody, I have some cool ideas to put in this proposal for the open call. And basically, with ASAP, you need to have a multidisciplinary project. And of course, Kerry, with all her amazing ideas, she came up with this Deep Connections project, which focuses on ecology and various things within that discipline. We have molecular, we have oceanography, biodiscovery, pollution, human dimension, so the social science aspect, and so much more. And of course, me being me, and the love of the marine realm, I of course jumped on this wagon and I said, okay, let's feed in a PhD within this proposal, and it got accepted. Yeah, got accepted in 2021. And basically, this was the first ASAP project that had a coelacanth driven research, basically, that is all focused on this guy. Okay, so of course, those of you that don't know what is a coelacanth, of course, coelacanths, they are from the genus Latimeria, and they are really rare. They occur in submarine canyons. They are not really accessible. And yeah, it's like basically trying to find a needle in many needles, okay, or a specific needle in a needle haystack. And they are basically the only extant genus of the ancient actinistian fishes. Basically, think about Jurassic Park, so but in marine, okay? So back in the dinosaur ages, and they are there's only two living species, which is the African coelacanth, which occurs around East Africa, in the West, in the West Indian Ocean, and then we have the Indonesian coelacanth. Alrighty. So what makes the coelacanth so special? Like, what's the whole fuss about this organism? So of course, coelacanths have these unique spot patterns that makes easy identification of individuals. They also have these pectoral and pelvic fins that move like in a tetrapod-like fashion. So think of a galloping horse, so moves. And then they also have lobe fins. They have these large reflective eyes. And then some other factors, 5-year gestation, and they have a really low metabolic rate. And just think about that for later on. So of course, looking at African coelacanths within our country, back in 1938, first live coelacanth was caught off the Columbia River near East London. Then only in the year 2000, the second location was found in Sodwana Bay, which was basically northern KZN. And then recently, a third location was found in the south coast of KwaZulu-Natal. So just look at the gaps. And basically, the National Biodiversity Assessment actually highlights the need for novel and non-invasive biomonitoring techniques for highly priority fishes or taxa. And for example, the African coelacanth is, of course, a perfect point in case. This species, of course, is critically endangered on the IUCN Red List. But now, how are we possibly going to do this? How can we accurately and non-invasively actually monitor the species. Okay, so now let me actually take you guys on a journey from dream to discovery. Okay, this is me. At the time, I was really young, only 31 years old. But yeah, it was my first time out at sea. I was super nervous at the start of my PhD. I was doing new, how do I say, I've been doing genetics since 2015, but I was doing everything new in my PhD. Didn't know how to filter water, nothing. Luckily, when it was with me, so I had some assistance. But anyways, as you can see, I was very giddy. But yeah, it was just, I was excited. Okay. Now, some of you will know that South Africa has about 20 odd marine protected areas. And for the purpose of this talk, I'll be focusing on the largest one, iSimangaliso. iSimangaliso was declared a World Heritage Site in 1999 by UNESCO. And iSimangaliso comes from it's a Zulu word that comes from miracle and wonder. And man, oh man, it is truly the embodiment of this word, and I will tell you now why. So, yes, iSimangaliso is home to coelacanths, but they also have an amazing rich biodiversity within its waters. And little did I know, on my very first day out at sea, I got to witness African coelacanths that many dream of seeing. And like I said, with the unique patterns, over here we've got Eric, and this one over here is Tor, and that one over there is Barbara. Don't they look gorgeous? But now, how are we actually going to do this? And environmental DNA might be a possible solution. So everybody has been talking about this whole eDNA thing. So what is eDNA? So eDNA is, of course, eDNA is, of course, DNA that's shed in the environment. Just think about this. Each one of you leaves some of your genetic material wherever you go. So if I have a reference database of everybody's genes or DNA, I can basically say, okay, Gwyneth was in here, was in this very room. Okay, so just think about that. Yes, we do leave a lot of our material behind. But anyways, so of course, we collect an environmental sample, in this case, water. And now what are we going to do? How are we going to get the DNA? So of course, we're going to extract the DNA back in the lab. With eDNA, you have to be extremely sterile, careful, meticulous. You cannot breathe when you're doing anything because it will end up in your sample. Extract the DNA, and then of course, what you have to do, you actually have to analyze and amplify the DNA. And we do that by using a photocopying machine, which is basically a thermocycler, a PCR machine, and you basically make millions and millions of copies of your DNA. And then what do you want to do? And by doing that, you either target a metabarcoding approach, which you can, of course, I want to target the fish in this, say, for example, in this environment. I want to say, okay, what fishes occur here, just looking at the environmental sample. You can, of course, look at, I don't know, corals or whatever you want, depending on the type of primates that you're using. Or you can also do a species-specific approach, which is a targeted approach. So, for example, in this case, we were looking at, can we detect coelacanth environmental DNA in water? Okay. And in order to do this, our amazing ROV pilot, Ryan Palmer, actually piloted on this expedition a targeted water approach. So what he did was, these are called Niskin bottles. These are two 1.75 liter Niskin bottles. We of course can't use the bigger ones because of course it's going to weigh the ROV down and it's going to make it difficult for the ROV to fly or maneuver. So anyways, what happens is basically he uses the ROV's manipulator arm, you see, and to keep the Niskin bottles open. Once we get down to the desired site, he releases the manipulator arm and closes the bottles. And he can continue doing all the other ROV surveys that's part of the project, looking at the fish community, looking at marine invertebrates, etc. So you can still continue doing the other components of the study. So of course, with our water, I was using, like I said before, the ROV, but I was also using, in some of the sites, I was using the CTD with Rosette. And then this is, of course, my filtering station. So, of course, I get my water, put it in sterile Nalgene bottles, and basically these little things are called Stereovex filters. And basically, it's filtered, the water is filtered through a vacuum pump. Okay, and then the DNA is trapped in here. Basically, all the genetic material, and then I use that in the lab, and I try and remove the DNA from there. Righty. So, this component of how to actually pilot the detection of coelacanth eDNA, we looked at 3 stations. So we have Jessa Canyon, which is a known coelacanth habitat. And then over here, we have 3 kilometers to the north, we have a deep slope over here, so open water deep slope. And then to the south, about 5 kilometers, we have another deep slope. Over there. And for intersect, you can see over here, this is Right Canyon, also known coelacanth habitat, but this is basically a coelacanth sanctuary. But anyways, you can see how small Jessa Canyon is compared to Right Canyon. Yeah, some of these things. But anyways, so in order to do this, we of course needed a species-specific primer. So the amazing Dr. Peter Schum from Liverpool's John Moores University designed a coelacanth primer, which is about less than 200 base pairs in length. And then what I did was amplify the DNA and then sequenced it on our very own Illumina MySeq here at SAIAB. Well, not me, Gwyneth. And yes, basically, yes, we actually found coelacanth DNA in Jessa Canyon, which is not really that surprising. But what was surprising that we actually found coelacanth DNA in our water samples collected by CTD down south. And as you can see, it's not a canyon, it's not a cave, nothing. And this paper it's currently in press. So please do look, if you want to know more about the study, you can see it in biology letters once it's okay. Now basically zooming out and looking at the fish community. So again, I'm looking again just at Jessa Canyon. So I'm maybe going to tell you about some of the fish that we detected. Are they friend? Are they foe? Or are they food? Righty. So again, the same water samples that I collected for the coelacanth component, we use the exact same for the fish community component, just using a different primer set. So we use the universal primer spanning the 16S region. And also to circumvent the challenges with filtering large volumes of water, but also to complement the water, we actually used sponges. But how can you use sponges to actually detect fish DNA or fish eDNA? So Professor Stefano Mariani from Liverpool's John Moores University and his colleagues, they basically published a paper back in 2019 where they actually looked at the fish community within a marine environment by collecting sponges. And basically this comes back to the basic biology of a sponge. They are sedentary, don't move, and they filter thousands and thousands of liters per day. As you can see with the scuba diver, they're basically using a fluorescent dye just to show how quickly a sponge actually filters water through the orifice. So you can imagine we have all this water and it's basically collecting like a DNA sampler, collecting a whole lot of fish, possibly a whole lot of fish in its DNA, in its tissue. So, of course, with the sponge sampling, again, targeted sampling using the ROV to be minimally invasive. So we use the ROV manipulator arm. This is Ryan. collecting a sponge for us, and then of course we bring it up on board and I preserve it. Righty. Okay, let's see, is it working? Okay, so this is the diversity within the water samples. So you can see we have some spadids, all these other things. Sorry, I'm not very clued up on all my fish. But yeah, so we got some of these. So I can just show you some of the spadids. I picked up some of them. Of course, we have some unknowns with environmental DNA or metabarcoding. The limitation is basically the reference database. So just think about, you're going to Pick and Pay. There's various types of sugar from different brands, but if it doesn't have a unique barcode, how is the teller supposed to say, okay, it belongs to this and this is the price? So of course it's going to match with a whole lot of unknowns, and there's lots of unknowns in our database because we lack the barcode. So we do need tissues. You can come to me if you have tissues that have been ethically collected, and I can try and update our database. So we're trying to grow our database, continuously trying to update our database. Yes, there we go. Then, of course, the sponges. Again, so as you can see, the sponges had a much greater diversity than the water. Again, yeah, so you can just see that. Not going to go into it. Alrighty, so zooming in on each individual biological sample, and in this graph, the blue is the water, and this beautiful color is the sponges. And as you can see, the species richness within each of these biological samples, overall, the sponges actually detected a lot more species. And for example, sponge number 14 collected about 66 species within it, and I only extract about 100 milligrams of tissue. So it's really tiny little piece. Okay, and this is again reiterated in the Venn diagrams, looking again at species level. So water detected about 38 species, sponges 49, and there's of course an overlap where there's about 11 species that was detected within the water and the sponges. Okay, so looking at this overlap, I'm just going to show you a few. So we have some slinger, it's considered food and a potential competitor as well. We have striped grunter, which is food, they say. One striped goldie, I absolutely love this image because they are a potential food source, and you can see this cutie, like looks like it's chasing the one striped goldie. And you also have yellowtail snappers and so much more. Oh, and a rock. Then looking at the water, we have Englishman, we have scorpion fish, John Dory, etc. Sponges, we have a sand smelt. We have, I didn't put the species here, but we have picked up, the sponges actually picked up 3 different types of suckerfish. It detected quite a few lanternfishes, so there was a lot of, I'm not even showing it in here, they detected quite a lot of pelagic fish. And then basically looking at the sponges, there were some mysteries that it actually solved. So Katie was puzzled by, Professor Katie Singh, she was puzzled by these fish, okay? And for the longest time, she didn't know what it was. First, she thought it was rainbow runners, but according to the fish eDNA within the sponges, it actually came up with this ruby fish. And when she actually, I sent my list to her, and she checked some of these species, and she said, cool, definitely seems like a ruby fish. And then another mystery was solved. for the fellow taxonomists. Apparently, rattle species, are quite difficult to actually identify, especially looking at just ROV images or surveys, and not actually examining the entire specimen. The fish eDNA within the sponges came back with a soft-haired rattle. So basically what we're trying to say is the usefulness of genetics, that yes, it's not something that will, I don't know, it's a good complementary tool. And this is basically just the tip of the iceberg. There is so much more data, still currently analyzing a lot of data. So there's a lot that we can still identify or explore, look at some of the species that require conservation or update the conservation management. There's maybe new distribution records or whatever, records, occurrence records. There is just so much more. There's a wealth of information that we are sitting on that we still, currently busy with. So basically, what about the future? So of course, I want to see, looking at the coelacanths, can we detect coelacanths in some of the other locations within the sampling region? I want to look at which of these sponges. Of course, there's been a lot of studies after Mariani et al. Published their papers. There's a lot of papers that look now at the use of sponges for eDNA metabarcoding of fish. But within the South African context, we want to see which of our sponges are better fish DNA samplers. And then, of course, looking again at the coelacanth, yes, we know the current distribution, which is in iSimangaliso, and like I said before, that recent one that was caught, not caught, sorry, why did I say that? That was observed in Mzumbe. But what about that live specimen, that live fish that was caught in 1938? Was it really a stray individual that people previously believed? Or is there actually a viable population that's just hiding in plain sight? We don't know, but we definitely want to explore. And basically, I just want to reiterate that as South Africa, we do have the infrastructure, the skills, the expertise to actually be at the forefront of cutting edge research. We can lead coelacanth-driven research with our own resources. This project was an amazing project. There's pollution, like I said, Dr. Sean Porter at Ori was looking at pollution. There's Professor Rosie Dorrington that's looking at biodiscovery within the sponges. There's oceanography. There's just so much more that we have access to because of, facilities like NRF with the SAIAB, and we have the stuff to actually do research. And even if you do not have the capacity, we can capacitate you. We can help you do your research. So please do apply. I just want to thank all these organizations. Also a special shout out to Megan van der Bank from SAMBI. She was an amazing integral part of the project. Just want to thank everybody, and thank you for listening. Any questions? Do I know?
Are there any questions?
It's just one of the heads for now. Just open the chat.
Yeah, thanks, Jody. That's very cool. Jody?
Yes, thank you. Sorry. Sorry, Ant.
That was very cool, thank you. I was interested about the ecology of sponges and potentially then linked to how you standardize. It's like if you're using those ecological filtering systems as how you collect your data, how you standardize sample volume between different animals. So one question is how much do we know about how long it takes for sponges to process or assimilate the DNA that's going through them? And then is this kind of consistent between different species of sponges and different sizes of sponges and flow rates in the environment? So just around like, what is the scope for standardizing sample size and sampling volume using sponges or other types of passive filtering animals?
Very good question. So basically, I do know that Professor Stefano Mariani at Liverpool, they have a whole sponge eDNA project where they look at where should you actually, which part of the sponge you should extract, but how I standardized it, because we didn't know at the beginning of the project, we didn't know what type of sponge we were going for. So we just chose any sponge. And basically I standardized it by only taking 100 milligrams of that tissue sample. So that was the only thing. So I don't know, at the end of this project, we will probably see which of the species or types are better DNA samplers, I guess, and then show people that, okay, going forward, if you do want to go this route, look at this, exclude, I don't know, glass sponges or, I don't know, encrusting sponges is not going to give you much. So haven't analyzed the data yet, still busy with lab work, guys. But yeah, that's the only way we standardized it, was just I only took a small piece that I extracted, so that I can say, okay, what was detected in that 100 milligrams? And of course, of that, you only take a small amount to actually amplify in the PCR process. But yeah, I tried to standardize it across my analysis, downstream analysis. But yeah, I won't know until I have my full data set.
Just also, sorry, has anyone worked out how long like the organic material lasts before it is digested by sponges?
I think there is studies, haven't checked it yet. Yeah, so there's actually quite a lot of studies. I did a quick glance. There's studies that's looking at, they compare, do a whole comparative study of using the ROV footage, water versus sponges. So there's actually quite a lot of sponge research now. Yes.
From Melissa.
Melissa, okay. Hi, Melissa. What do I do now? I'm muted.
She's going to unmute now.
Okay.
Let's not get to unmute.
Hi, Melissa, can you please unmute? Unmute.
Jody, my laptop is not read the comments.
Okay.
Yeah.
So the question is, were you able to identify the various sponge species in those waters and the vertebrates that would use these sponges as residential motels?
Okay, let me just read that question. Okay, so I am still currently analyzing that. We are barcoding all the sponges that we collected.
Sorry, Jody, do you have a power cable?
Oh gosh, sorry.
Sorry, just give us a second. Her battery's about to die.
Sorry,
That's where
he's plugged in. Yeah. Sorry about that. Don't know why it's not. Is that actually?
There we go. Oh, no, it's not.
Yeah, I think it's just a. Oh, golly gosh. Sorry about this. Okay, thanks, Lucky. So yes, Melissa, I am currently barcoding all the different sponges, and then basically from that, and once I have my full data set, I will see which of these species are better DNA samplers. And then is there anything else? What is this?
Just using the mammals.
Yes, oh, if you want to look at invertebrates, of course, I was only looking at the fish. If you want to look at invertebrates, it depends on your primers. So if you want to look at corals, you need to use coral specific primers. If you want to look at, I don't know, any type of, or for example, if you just want to look at sharks, you can make it species specific. So, or genera or family or whatever. So it just depends on the primer that you're using to actually amplify that DNA. Okay. I hope that answers your question. Any other questions?
Rosalind has a question.
Oh, hi, Rosalind.
Hi, Jody. Thank you so much for a great talk. That was really interesting, and your work is incredible. I can see a lot of useful stuff coming out of this. Just for interest's sake, so you mentioned that your primers are coelacanth specific. So are you just able to detect presence absence, or are you actually able to distinguish between, does it give enough data to distinguish between the individuals like the spots do? You're laughing. Has that been asked before?
Don't worry, I got that question a lot.
Space.
Yeah. Let's watch the space. At this point in time, I cannot tell you if the primer is variable enough to actually pick up intraspecific variation. So can it tell between individuals for the general public? Can it tell between that, or can it only say, okay, this is a coelacanth, African coelacanth? Yes or no?
Hoping it's not that.
Yes, we're hoping, but we will see once we have a complete data set.
Oh, cool. I look forward to the answer. Good luck.
No pressure.
No, none at all.
Yes.
Albert?
Okay.
Yeah, thanks, Jody. I really enjoyed your talk. You had 3 localities where you collected your samples for the coelacanth.
Yes.
And I maybe, yeah, if I got it wrong, you can correct me. I thought the other one was a negative control, and then the other one was a positive control where you know that they do okay, or they've been observed in the past. But then you recorded in the southernmost point.
Yes.
You also found
Yes.
presence there, which I assume is a false positive, or maybe can you just clarify that?
Okay.
Yeah, thanks.
So basically with the coelacanth sampling, we of course collected in known coelacanth habitats such as Jessa Canyon. And then of course in potential coelacanth habitats, whether it's steep slopes or unknown. So basically the northern slope was basically a deep slope that 3 kilometers north of the known site, and then the south. So we basically, it's a deep slope. We did not know. It has no, there was prior to collection, there was ROV dives just to see. So it's not a coelacanth. Doesn't look like a coelacanth habitat. Very sandy, salty. Yes.
So we don't know if they move between canyons.
Yes. Yeah. So basically there was a study done that showed the, is there any way that I can go back? How do I do that?
Yeah, just share.
Share my screen. Okay. So where do I go? Oh. That was just gonna be the longest. Expected. But basically that large canyon that I showed you, Right Canyon. So in Hiemstra et al. 2006. Let me just get a clear picture. Oh, yeah. So Right Canyon and then Jessa Canyon. So there has been evidence or been observed that a few individuals or 1 or 2 individuals have been known to actually transverse these grounds. So even though this is not a coelacanth habitat, it's not a cave, it's just a deep slope. I think the tagged individual was actually, I speak under correction, was actually found or observed based on the tracking data that it actually moved between Jessa and Right. Over here down south, absolutely don't know. And this is not a false positive. Based on all our field controls, I also noted it's based, it's in the paper. We extract and we analyze each site separately to reduce contamination. We don't, I work in a different lab where there is no coelacanth present. So of course, I don't do my work at SAIAB. So extract all that stuff. I have extraction, I mean, I have controls, all my controls were clean. So it is not a false positive. But of course, at the time of collection, because we collected with CTD water, there was no visual confirmation, because of course CTDs doesn't have a camera. But yes.
Thank you.
Any other questions, Karen?
Jody, kind of following on from what Elbert was asking, how, I don't know what the right word is, but like how good does the DNA stay in the water for? So if it's flowing with currents or something, is it going to be viable for a week or something? I'm just thinking of like current movements and everything. So with that south sample, I'm not saying that they get everything on coelacanth there, but that habitat was like, so is it a case of that water shifting down and that DNA being carried down into that area? Is that a possibility?
So basically there's been studies that looks at the decay of DNA within the water, and basically there is a significant drop within 24 hours. Yeah.
So part of the world of current screens, yeah?
It does. So it's not beyond the realm
Yeah,
of
yeah,
possibility,
for sure.
but
I guess if there's quite a lot of coelacanth, there should be quite a lot of DNA. Well, lots of coelacanth, but
yeah,
quite a lot of DNA in water.
also
Yeah, it's fine. Continue, supervisor.
all of the samples we took in JSA, we took multiple samples, not all of them came out positive.
Yeah.
Okay, so where we could only see one coelacanth, we weren't picking up a lot of DNA. We needed 2, 3, 4 coelacanths in order to pick up that DNA.
Yeah.
And that's probably because of the low metabolic rate of the coelacanths. They don't move a lot, so they don't shed a lot.
Yeah.
So the fact that we aren't picking up throughout JSA, we know there are coelacanths, but we are picking up downstream, indicates more likely that there was a coelacanth there rather than wash down of the DNA, because if it was just washed down, we would have picked it up in all the other samples in between that southern site and JSA and also along JSA.
Thank you. Anything?
Who has a question? Release, I just want to know if there were any invertebrates on the sponge that you saw, and Kerry answered some squat lobsters and rock lobsters.
Thank you, Katie. Yeah, I think is that okay? Yeah, that's it. Good.
Was there no other questions?
No other questions? Yes, please. Yeah, be a lookout for all the other exciting research that will be. Yeah.
As Jody said, this is only part of her thesis. She has a very ambitious thesis. This is only one section of it.
Yeah.
And this data set's not 100% complete. It'll be complete by the end of the year.
Oh, yeah.
And more papers, watch the spots. It's going to be very exciting. And I'd like to say thank you very much, Jody, for a lovely presentation and for giving us a good overview of what you're up to and the cool stuff that you're involved in.
Thank you. Thanks. Thanks, everybody.